The overall goal of this procedure is to generate genetically modified organotypic skin cultures using devitalized human dermis to determine the impact of altered gene expression on epidermal growth and differentiation.
This method can help answer key questions in the dermatology field, such as determining which genes are responsible for epidermal growth, differentiation, and progression to disease. The main advantage of this technique is that genetically altered human skin is generated within a few days, which allows the study of human samples that are potentially clinically relevant. Demonstrating the procedure will be Jingting Li from my laboratory.
If receiving frozen human skin, place the tissue in the tissue culture hood for 3 to 5 minutes to thaw. Once thawed, transfer the tissue into a sterile 50 milliliter conical tube or 125 milliliter bottle, depending on size. Next, add 4x penicillin and streptomycin in 1x PBS until the container is 75% full. Cap the container and shake vigorously for 5 minutes. Remove the antibiotic solution and repeat this step 3 more times. After the last wash, transfer the tissue to a new container and add fresh antibiotic solution. Incubate this mixture in a tissue culture incubator at 37 degrees Celsius for 2 weeks to allow the separation of the dermis from the epidermis. When 2 weeks have elapsed, use forceps to peel away the epidermis from the dermis. Discard the epidermis according to the institution's protocol for dealing with